Hi all, thank you very much for tuning in with my channel, the channel in ozone technology. This session will be session 9A, which will be predominantly fundamental biology applied in ozone technology. The topics I will be introducing in this session will be the basic cell structure, the mode of kill by biocides and ozone, the structure of fungus and cysts, and the mode of kill, the basic viral components or components of the virus, and the method of inactivation of virus, and some information of smells and odors. In order to understand how ozone works as a biocide and how the biocides, normal biocides we use, also destroy or kill the bacteria, we have to know a little bit about the structure of the cell. There are two types of cells. One is a plant cell and one is an animal cell. In the case of a plant cell, the cell wall is very thick, while in the case of an animal cell, the cell walls are thinner. Now, I will go into the cell wall a little bit later, but within the cell, you have certain components. If you look at the diagram I've given you on the right of the slide, and I'll go in the clockwise, you have the mitochondrion, you have the Golgi apparatus, the nucleus, nucleolus, centrioles, cytoplasm, which is the contents of the cell, microtubules, which gives some sort of structure of the cell, the ribosomes, which takes part in the manufacture of proteins, etc. The plasma. Plasma is the one that has got all the nutrients, components that carry the oxygen, carry food materials, etc. Then you have the cell membrane or cell wall, we call it in plants. Then you have the endoplasmic reticulum, the two types, the vesicles, Golgi vesicles, very similar to the Golgi apparatus, the lysosome, and finally the pinocytotic vesicle. That's through which a lot of food and other components may enter the cell. Now the nucleus will contain nuclear proteins, that is the RNA. The RNA is also found in the cell itself, as I did mention. DNA, the nucleus itself composed of the DNA, and the cell contains, plasma will contain the RNA and the tRNA and mRNA. Now coming back to our cell membrane, there is a difference between there is a difference of electropotential between the inner of the cell membrane and the outer of the cell membrane. It may be between minus 80 to 20 millivolts. The cell maintains its life by keeping this electropotential difference, and in order to do so, there is a sodium pump which pumps out the sodium and allow potassium into the cell to maintain the specific ratio to achieve the volt difference. In other words, if the voltage difference is disturbed, the cell will cease to exist. So that is extremely important to maintain the life of the cell. The diagram in this slide shows a blown up picture of a membrane. You find the membrane consists of chemical or protein called phospholipids. So it's a phospholipid layer. It is bound, two layers are bound to each other to form the membrane. As I did mention to you, it is necessary to keep the voltage difference facilitated by the sodium pump so that the cell is kept alive. Plant cells are also work in a similar capacity, except they have a thick cell wall. The fungus and a few other cells also have got the very similar structure. Fungus is a class of organisms that are a bit higher than the bacteria. Bacteria generally are single cellular structures, whereas fungus can be a series of cells joined together, or you call them multicellular chain of cells. The chain can be simple chain or branched. A good example of fungus is yeast or mushrooms. Though they may be multicellular, the cells are equally exposed to ozone and may be susceptible to ozone, unlike layered multicellular tissues in the higher animals, which are less susceptible to ozone. The fungal spores are unique, and the fungi are almost also susceptible to ozone. You must remember, ozone attacks single cells and loose multicells. But if they are composed of very high number of multicellular organs, then the ozone in its short time of action will have almost no effect on the organs or the multicellulars. Cysts are actually microorganisms that develop a very strong, tough, protective layer around itself. And they are a bit hard to kill with normal concentrations of ozone or any other biocides or bactericides, but can be killed when ozone is applied at higher concentrations and in concentrations with combinations with other oxidative chemicals. There are three types of biocides. One is oxidative, the other is non-oxidative or metabolic, and the third is radiation, such as ultraviolet. So non-oxidative biocides are generally metabolic, whereas radiation and ultraviolets are electrical energy or radiation energy. Non-oxidative biocides are, for example, antibiotics or chemicals that can cause disruptions in the cell metabolism and kill the cell. Almost all of them are organic and attack certain aspects of the metabolism of the cell. So between the oxidative and non-oxidative, there is a big difference. One oxidizes, the second, metabolic or antibiotics, interfere with the chemical reactions within the cell and stop the reaction, and thereby prevent the cell from continuing its functions. Let us consider non-oxidative biocide. Here we shall consider an antibiotic. Antibiotic works, as I did say, diffuses into the cell bacteria and it interferes with the critical metabolic processes, preventing the reactions from continuing and starving the cell to death. Now, effect of the antibiotic is both time and concentration related. When I mean by time and concentration related, it means that from the time you apply the antibiotic, it has to first diffuse into the cell, bacterial cell, and then we'll have to select specific reactions within the cell, what the antibiotic is supposed to or designed to do, and prevent that particular reactions. When those reactions are interfered, then the product of the reactions being very critical for the cell will not be able to continue surviving, and therefore it will virtually starve to death. Another effect of the antibiotics is to interfere the cell multiplication. That means the chemical process of multiplying the cell is interfered by the antibiotic so that the cell will not multiply, which means the cell will have to live out its normal life and die after its lifespan is completed. Bacteria that survives the antibiotic through the inefficient application of the antibiotic can modify their structure or its reaction characteristics and develop resistance to their antibiotic. This necessitates the modification of the antibiotic regularly to meet such resistance characteristics. This is an ongoing phenomenon. The next form of biocide is radiation and ultraviolet rays. Ultraviolet rays are very high frequency electrical or energy waves. They tend to have a very similar effect as ozone in the mutation of the nucleus of the cell and cause disruption in the control of the metabolism and cause death. Application of ultraviolet radiation is described in detail in later presentations. Now we have come to the end of this session 9A. I thank you for having the patience to listen to my presentation. please go to the next session 9B, where I will be discussing more on oxidative biocides and some information on smells and odors. If this is your first tuning, please visit sessions 1 and 2 that will complete the program details of this course, and sessions 3 to 8. Also, please click the like icon and the subscribe icon, and I will be able to notify you as soon as I uploaded the next session. Thank you and have a very good day.